Okay, I hope you see it in full screen now. And I'm going to start thanking the organizers for inviting me. Thanks, Julia and Letizia. The workshop has been great so far. I'm sorry that I couldn't make it to Rome because of COVID, but yeah, I'm here. Thanks for giving me the opportunity to present online. Okay, so today I'm going to talk about computational approaches to study cortical dynamics at multiple scales. So I guess I don't have to convince you at this point, why do we want to study cortical dynamics at multiple scales? Well, of course, because brain dynamics is a cornerstone of human consciousness, and indeed brain dynamics varies continuously over time, both under physiological and pathological conditions, showing different spatiotemporal features, as has been already shown in the workshop extensively. And so it is important to study these at multiple scales because different scales of observation may provide different information about the brain functional mechanisms. And so computational tools are needed in this context to answer our scientific questions about brain functions and make sense of all the data that we have acquired so far and that we still record every day in the labs. Where in eBrains we have a great example of a collection of data from different species and different scales that can be analyzed by the users through the use of computational tools. So today I'm going to talk to you about 3 computational methods that can be used to provide a comprehensive characterization of brain dynamics and of their properties at different scales and in different brain states. So the first is the study of waves propagation, network synchronization, and cortical irreversibility. So these methods can be applied at multiple brain states, ranging from the asynchronous awake state to the synchronous slow wave activity characteristic of the anesthesia states and slow wave sleep. But as well, they can be applied to different scales, varying from micro to macro scales, taking advantage of the use of in silico, in vitro, and in vivo models. So I will show you the application of these 3 methods in different subsets of this space. So wave propagation is a method that can be applied both in vivo, in vitro, and in silico in under conditions, dynamical conditions dominated by slow wave activity. So during slow wave sleep or under anesthesia in vivo. While network synchronization is actually a method that can be applied to all the different brain states that we have here, and also at multiple scales, ranging from micro to macro scales. And finally, the cortical irreversibility is able, I will show you that this is a method that is able to discriminate all these different brain states, both at whole brain level and at the level of single areas. So let's start with the waves propagation. So here we developed and validated a novel method to study how the cortical events, in our case, the up states of the slow wave activity that you already saw during the workshop, how these events travel across the cortex as propagating waves. So this method relies on the computation of the instantaneous phase of each network node through the use of the Hilbert transform. This approach transforms a real value time series, so the signal that we record, into a complex phasor, whose modulus and argument in the complex plane represent the signal instantaneous amplitude and phase. So we computed the phase of the broadband multi-unit activity in order to preserve its spectral content and to avoid distortions or artifacts that can be introduced by filtering. And the use of the phase in this context is more reliable than the use of the amplitude, especially in noisy recordings, in electrophysiological recordings. So we detected here the waves in the multi-channel recordings as the phase latency to a given phase crossing. So this creates the phase latencies that give rise to the shape of the spatiotemporal profiles of the waves traveling across the network. So thanks to this method, we were able to reconstruct the patterns of propagation of upstates in different cortical preparations at different scales, so both in vivo, under anesthesia, in vitro, and in silico. So our results show the emergence of two clusters of propagating waves in the three preparations. And also, we show some differences, of course, across scales. For example, about the speed of propagation that is slower in vitro than in vivo. But overall, what we show is that the main mechanism related to the generation and propagation of the waves in these three cortical systems is similar at these three scales. So, as I told you before, this method is quite limited to be used in slow wave activity conditions because it relies on the detection of these waves in the multi-unit activity signal. And also this method needs an a priori assumption about the number of modes of propagation to be found in the data set. So in order to free ourselves from this constraint, we also developed a new method for the unsupervised classification of waves patterns that we called energy-based hierarchical waves clustering. So these methods, to develop this method, we depart from the idea that the brain may be seen as a nonlinear system outside of equilibrium. And here we introduced a new computational tool based on the modal analysis of fluid flow, which can be applied to electrophysiological data. So this tool is based on the application of an empirical eigenfunction approach for dimensionality reduction, which is the singular value decomposition. Thanks to this, we create a three-dimensional representation in which we project the network activity in the space defined by the first three spatial IM modes computed through the singular value decomposition. This is a video showing you an example of how the cortical activity in vivo, recorded in vivo, this is 25 seconds of activity, can be mapped into this space. So here you can see that the points develop in time as low dimensional trajectories, and the shapes of these trajectories described by the waves while traveling across the network are what we are going to use to classify our waves. So we collect a set of vectors describing the trajectory associated to each activation wave, and we compute the Euclidean distance between these trajectories, obtaining distance matrices. These are three distance matrices that we obtain from electrophysiological data recorded in vivo under three different anesthesia levels: deep, mid, and light anesthesia. And you can see that here we can clusterize and so classify our waves, clusterize the matrices using a hierarchical clustering algorithm, which is unsupervised and provide us with a number of patterns contained in our data, and also with an idea of how rich is the spatiotemporal dynamics of the cortex under three different conditions. So I presented you methods that can, in this case, be used to study the spatiotemporal patterns generated by the cortex and how these are related to the dynamical state of the network. So while wave propagation reflects differences in phase across the network, the relationship between the phases at different network locations may provide also a measure of network synchronization. So in order to study the network interactions occurring at different scales, we computed here the evolution of network synchronization over time, estimated by the Kuramoto order parameter. And we studied here the synchronization dynamics at two different scales. So both in the local field potential that can be seen as an input signal. It is an extracellular signal generated by the transient imbalance of the ionic concentrations in the space outside a cell, and that results from the cellular electrical activity. So the input signal, but also in the output signal between neurons, we can compute the synchronization of the multi-unit activity that can be seen as the output signal because it is mainly made from the output action potentials generated by the neurons postsynaptic activity. And so interestingly, we know from previous studies that these two components that are actually simultaneously acquired by the electrophysiology, they may bring different information about network dynamics. And indeed, here I'm showing you how the input and the output synchronization actually bring different information about the network dynamics. So I'm showing you here network synchronization through a dynamic phase histograms computed both on the multi-unit activity here on the left and on the local field potential. So in these histograms, the color scale indicates the number of electrodes with a given phase at a given time. And then in the middle panels, I show you the Kuramoto order parameter, and in the bottom panels, I'm showing the corresponding activity of each node of the network or recording electrodes. So this is at multiple scales, in silico, in vitro, and in vivo. So from these dynamic phase histograms, you can see by eye differences in the synchronization dynamics of the multi-unit activity and local field potentials, supporting this idea that these activities, two components, actually bring different informations. So on one hand, we have in the multi-unit activity a kind of synchronization, the synchronization characterized by a regular behavior where we see a prevalence of in-phase locking during the up states and a synchronous stochastic behavior during the down states. On the other hand, in the local field potential, we found a different behavior and actually a very interesting behavior of synchronization during the down states. That is what we call the metastability of the down states. So metastability here is characterized by two phases. The first is a sustained period of high synchronization, which occurs right after the up to down transition, where the collective phase behavior shows an exponential trend and the Kuramoto order parameter is close to 1. Then this period is eventually followed by a second period characterized by poor network synchronization, in which the collective phase randomly fluctuates between P and minus P. And for this reason, we named these two stages the deterministic and stochastic stage, respectively. So according to the state of the network, one stage may actually prevail over the other. And also, interestingly, we observed that this deterministic stage is very much dependent on the strength and on the duration of the after hyperpolarization mechanisms, and so on the state of the network that modulates the adaptation dynamics. So in order to confirm such observation, we used our in silico simulation. So for each metastable down state, we were able to isolate the exponential branch of the synchronization, which characterized the deterministic stage, and to perform an exponential fit on each of them in order to estimate the time constant of this exponential. And we demonstrate here that the time constant estimated from the exponential phase synchronization is highly correlated to the time constant decay tau of the adaptation current that we impose in the model equations. So this means that we reveal here the possibility to infer a microscopic phenomenon as the evolution of the after hyperpolarization current following the up states through the study of a macroscopic variable that is network synchronization. So this is an example at the microscale in silico, but we of course wanted also to apply this method to different scales, so also in vivo and in vitro. So we provide here a dynamical representation of the two systems based on the kernel density estimation of the distribution of the downstates duration versus the tau, this time constant that we estimate. And in order to disentangle the dynamical differences between the in vivo and in vitro cortical networks, we also used here experimental manipulations to modulate the cortical activity, both in vivo using anesthesia, using different depths of anesthesia, and in vitro using bicuculin methionine, which is a GABAergic blocker. So we observe here that the relationship between the downstate duration and the adaptation characteristic time constant becomes increasingly linear when moving from deeply unconscious brain states under deep anesthesia towards lighter anesthesia states. And the same somehow happens in vitro when we applied increasing concentration of bicuculin, and we enhance gradually the after hyperpolarization drive on the cortical dynamics. So this, again, is an example of a method as network synchronization that can be used at multiple scales and under different network conditions, providing information that cannot be inferred by looking at a single scale alone. So last, I'm going to present you a method to study cortical irreversibility, and I'm going to show you how this method is, this measure is actually closely related to cortical dynamics. So again, the cerebral cortex as a whole is able to express different dynamics according to the brain states. And in this context, the transition across different brain states can be seen as generated by the dynamical evolution of the brain system that unfolds towards and away from its equilibrium point. So accordingly, the degree of non-equilibrium of the macroscale cortical dynamics that is measured by the temporal asymmetry of brain activity can be used to discriminate different brain states and also to characterize the cortical dynamics at different scales. So following this framework, this idea from Gustavo Deco, here we quantified the temporal asymmetry of electrocorticography signals recorded in chronically implanted rats at whole brain, so in both hemispheres in different regions, during fade-out anesthesia protocols and also during the natural sleep-wake cycle of the rats. So the reversibility here is computed under different brain states, under all these different brain states that we can detect during the fade out of anesthesia and during the sleep-wake cycle in 10-second windows that are extracted from the time series recorded from different cortical areas. So let's consider two time series recorded from two different cortical areas, X and Y. We compute here their reversed backward version, XR and YR, by flipping the time ordering of the time series, as you can see here. Then we measure here the causal dependency between the two time series, both in forward and reversal, by computing the correlation between X and Y shifted in time by a time step that here is capital T. So in this way, we obtain the C forward and C backward correlation matrices, and the irreversibility, I, here is estimated by the absolute difference between the causal relationship of these two time series, both in the forward and reversed time evolution. So using this method, we here computed the irreversibility, the pairwise irreversibility per brain area, but also the average irreversibility averaged across the areas per different brain states, and then the hierarchy measure or the variability of the level of irreversibility across different cortical areas. So Our results here show that a low degree of irreversibility is associated with deeply unconscious brain states, being that they closer to the thermodynamic equilibrium, and that this irreversibility progressively increases during the recovery of consciousness. So accordingly, wakefulness has been shown to be the state with the highest irreversibility, highest variability, but also the highest variability in space across areas, as you can see from these macroscopic maps. While deep anesthesia is characterized by the lower value of irreversibility and also by a very homogeneous distribution of irreversibility across different cortical areas. So these maps also highlight an interesting property, which is that actually slow wave sleep, even though it has a low level of irreversibility on average, it actually shows quite a good variability of irreversibility across different areas, which is totally different from what we observed under anesthesia. So even though the dynamical state is the same, slow wave activity, we observe this difference. And this may be due to the fact that slow wave sleep, first of all, it is a natural brain state. And so the cortex needs to keep a kind of a level of complexity, of diversity, in order to respond to, for example, the need of suddenly wake up. So this is, again, a method that can be applied, as you see, at different scales and also under different brain states. So to conclude, I showed you how the study of the spatiotemporal patterns of propagation of cortical waves at multiple scales can actually provide important information about the underlying network features and brain dynamics. That it is possible to infer microscopic features of network dynamics by studying macroscopic variables such as collective synchronization at multiple scales, and that the temporal asymmetry of brain activity, quantified by the irreversibility of the brain, can be used to discriminate different brain states, both during the natural sleep-wake cycle and during anesthesia, in rats, as I showed you, and in non-human primates, as Gustavo will publish very soon. So thank you very much for your attention. Thanks to the people who work with me in all these projects, Mavi Sanchez-Vives, Maurizio Mattia, and Andrea Galluzzi, and also Gustavo Deco and Morten Kringelbach. And thank you for your attention.